In this talk, Matt Dallas will introduce Essential 8 Flex Medium, a xeno-free PSC culture medium that eliminates the need for daily feeding without requiring very low seeding densities or other significant changes to current PSC culture protocols. Matt is currently a staff scientist in cell biology R&D within the Life Sciences Solution Group at Thermo Fisher Scientific, formerly Life Technologies. He obtained his PhD in chemical and biomolecular engineering from the Whiting School of Engineering at Johns Hopkins University, which included a fellowship within the Johns Hopkins Institute for Nanobiotechnology, a multidisciplinary institution with efforts spanning basic biological science, clinical science, and public health. Matt joined the Life Technologies Cell Culture Essentials Group in 2012 and has since acted as the technical lead on various cell biology programs involving oncology, primary human tissue culture, and stem cell biology.
Jen, thank you for that introduction, and everybody on the phones. Really appreciative of you giving us the opportunity to speak with you today. I'm really thrilled to have this chance. I've been looking forward to this for quite some time, for the ability to, in some cases, introduce, in other cases, reintroduce Essential 8 Flex Medium, which is one of the newest product offerings out of the Gibco family. So before we get started, I actually want to make a few technical comments. For those folks on the phone, you should be seeing a chat window where you can enter questions as I'm giving this presentation. Please feel free to do this throughout. We're actually going to have, hopefully, an active question and answer portion towards the end of the talk. But definitely don't feel like you need to wait. You can enter those questions in real time, and we'll see them pop up, and we'll curate them as we go through and save them all for the end. A couple other technical notes. If you do have any problems with not being able to see the slides or seeming that it's stuck, just try refreshing your browser. We've heard of some technical difficulties, and that seems to fix any that you might run into. And as a last note, I wanted to remind everyone that these slides actually will be, these slides and this talk will be recorded. So if you think you miss anything or you want to go back and refresh your memory, these will be posted online for your consumption later. And with that, I'll kick things off. So in order to get everybody in the right frame of mind, I thought I would start with a brief review of how human pluripotent stem cell culture has really evolved over the last 10, 15, 20 years. And so as many of you on the phone are aware, in 1998, the first publication detailing an optimized culture system for human PSCs came out of Jamie Thompson's lab at the University of Wisconsin. Now, for better or for worse, this system did rely on both FBS supplemented medium as well as a supportive feeder layer to maintain these rather finicky cells in culture. So it was a groundbreaking step and one that really laid the foundation for a lot of the spectacular work that's gone on since. But because of those limitations in the way the system was developed, it did make transition of the technology to other labs a challenge, as well as introduce some inherent variability due to both the feeder layer and the fetal bovine serum supplementation. Just 2 years later, there was a new report detailing an adapted culture protocol that replaced the fetal bovine serum with a Gibco product, namely knockout serum replacement. And now this system did maintain that supportive feeder layer, so while it was a great step in making a more defined medium, it still did introduce the finicky nature of those feeder cells, as well as it made use of a supplement that does still contain the animal origin components. That can lead to some difficulties in reproducibility in transferring the system between labs. Just a year later, there was a new publication detailing the ability to actually use feeder-conditioned medium in place of those supportive feeder cells. So it's, I think, a really nice step in simplifying the HPSC workflow and removing that feeder layer. But again, because we're dealing with now a really intensive biological system to create the media, you do, again, introduce some variability as well as some significant difficulties in manufacturing a reproducible product. Fast forward a few more years and we come to the period between 2005 and 2007. That was a very busy time in the human pluripotent stem cell arena as we saw the development and launch of the first few chemically defined media for these types of cells. You may be aware of the Gibco StemPro ESC SFM You may be aware of M-teaser, and there are some others as well. And because these media are all chemically defined, it made the formulation process much more reproducible. You had a little more consistency lot to lot. And it also did create a system that was a little easier to use, a little easier to transfer these technologies across various labs. Now, all those benefits aside, we are still dealing with supplements and media, therefore, that contain components of animal origin, in particular bovine serum albumin, which I think, as many of the people on the phone are aware, can exhibit, in some cases, significant lot-to-lot variability and do require, in some cases, that you actually do lot testing on these media. A few more years into the future, and we're at 2011, and this really highlights the launch of the Essential 8 medium system. Again, this is a Gibco product. It is another chemically defined media, but in this case, we're talking about xeno-free. So we've eliminated the use of some of those pesky animal origin components, namely BSA, and in doing so, smoothed out a lot of the hiccups that you can run into with, again, this fairly challenging culture system. So for those of you who are not aware, I did want to talk a little bit about Essential 8 medium and the benefits that it provides. So as I mentioned, it's a chemically defined, xeno-free, and obviously feeder-free medium system. And unlike some of the media that I just introduced on the last slide, Essential 8 was actually specially formulated for the expansion of human PSCs. In some cases, some of those other media were designed around other cell types and then tweaked, adapted to work with human PSCs. Essential 8 was, from the beginning, operating with human PSCs in mind. Now, Essential 8, you see the components, really the name speaks for itself. It's including really only the things you need to culture these human pluripotent stem cells. Not only have we removed BSA, but because it's such a lean formulation, we have virtually eliminated the potential for complex component-component interactions that often happen in some of the more complex, rich culture media. These interactions can occur either during the formulation process or in the process of your cultures, and either way can lead to instabilities and other challenges in the media that may make your results difficult to interpret. As is the case with a lot of the other Gibco products that we offer, Essential 8 Flex comes under Essential 8 comes under CGMP quality standards, so you know that every bottle you use is going to be as good as the last one. And then finally, a nice supplement to the Essential 8 medium family is a defined substrate. Here we're talking about the truncated recombinant human vitronectin. You can see the catalog number there. In combining this with Essential 8 medium, you have a fully defined system that's consistent, economical, and scalable. Now, all that being said, I think a lot of the people on the phone will agree with me here that routine stem cell culture still can be fairly painful. And for me, speaking in particular, those last 2 days in the workflow I'm showing here are the most painful points. If you want to keep your cultures as happy and healthy as they can be, you're really going to be best served by coming in and feeding your cells on a daily basis. And unfortunately, that includes Saturday and Sunday. Now, it turns out that the need for this daily feeding is largely driven by some key molecules that exhibit a loss of activity during exposure to elevated temperatures, really at 37°C. So without these key components, you really lose quality cultures, and that can be indicated by the slide, the image I just showed here. FGF2 is a good example. If you are culturing cells in the absence of active FGF2, you'll get cultures that look a lot like what I'm showing you here. You have maybe small islands that have that nice compact PSC morphology, but by and large, you see massive, irreversible, spontaneous differentiation. So these molecules are key, and unfortunately exhibit fairly low activity during prolonged incubation at the temperatures at which these cells need to be grown at. Now, in addition to this key issue, the routine culture that is required today for using standard PSC media does introduce some additional challenges. Now, every time you're feeding your cells, you're obviously taking them out of the incubator, you're perturbing them a bit, as well as opening up new avenues for contamination. So a potential issue there. And one other thing that we notice when we go out and we talk to customers to understand better how they're interacting with their cultures, what we find is that in order to cover the odd weekend off or long weekends, holidays, sick days, things like that, we find that multiple users are handling a single culture, and oftentimes through a series of critical experiments. And you know that due to differences in the way people are handling cells, just on a general basis, you may be introducing variability into your cultures. that can cloud some of the results that you're looking to find. As well as, especially if you're using folks that are not necessarily fully adept in this rather complex culture method, could be opening up new avenues of contamination there as well. So with these challenges in mind, the team, the Essential 8 Flex team, set out to develop this novel media system. And as I said before, I have the pleasure today of introducing you to Essential 8 Flex Medium. The goal of this product, and really the output, is going to be that it's going to enable you to take back control of your PSC culture schedule. I think in a lot of ways, I know personally speaking, sometimes my life gets designed around when I have to be in the lab and when I have to split my cells. And with Essential 8 Flex, we're hoping that we can reverse that, and you're able to fit your PSC culture schedule into your lives as they stand. Now, we really see this as the next step in the evolution of PSC culture medium. I hope that by the end of the next half hour to 35 minutes, you'll agree with us there. So Essential 8 Flex Medium, what is it? Well, it's a formulation that's based on the wildly successful Essential 8 Medium. As a result, you're getting the chemically defined nature, you're getting xeno-free, feeder-free culture with that GIBCO CGMP quality. No compromises on that regard. The added benefits that Essential 8 Flex brings to the table, however, is the fact that with the formulation changes that we made to that initial Essential 8 formulation, we've been able to optimize the activity of some of those key heat-sensitive compounds that are present in this media. And I'll use, you'll see throughout the talk, FGF2 as a good indicator of some of that sensitivity and activity. In stabilizing these key molecules, what we're enabling here is weekend-free culture, the long story short. So with the optimized workflow that I'll introduce on the next slide, what you're going to be able to do is eliminate up to 3 days of media changes in a single week, and 2 of those days off can be consecutive. And so that means weekends free. Not only are we reducing the time you're going to be spent handling your cells and also the regularity with which you're going to have to be handling them, there's also a reduction in volume usage. So about 30% reduced media consumption with this novel media and protocol that goes along with it. All the while, I think this is just as important to say, as the workflow changes, you should be expecting no impact on health or pluripotency of your cultures, and I'll demonstrate that in the extensive data set that we put together. This is true over very long periods. So here's the protocols that I mentioned. For those of you on the phone that are doing a twice-weekly split schedule, and from what we find talking to customers, that seems to be the majority, you may be targeting something like a Monday and Friday split. And in between, every day, you should be feeding your cells, getting the optimal result that way. But with Essential 8 Flex, we tried to adopt a similar workflow where we're maintaining that twice-weekly split schedule. Your cells are never getting split at a very high density. And actually, there are some protocols out there for commercially available products that do claim to support one-off weekend-free culture here and there. What we find is that these protocols really do rely on a single split, say on that Friday before the weekend, at a very low density. You can kind of squeeze your cells through those 2 days with no feeds, and then coming in the following Monday, you start feeding every day and waiting for your cells to reach the confluence level at which you can actually play with them again. And that's what we're really trying to avoid with this optimal protocol and Essential 8 Flex. So what we've developed is a protocol where you're splitting in a 7-day period on days 1 and 4, and you have a feed the following day, the day after you split. So 24 hours after you split, you feed. This should be compatible with folks that are currently using ROCK inhibitor, as you want to generally change the media 24 hours after you've plated the cells in the presence of that inhibitor. But in the instances where you're not using ROCK inhibition, we still suggest doing that 24-hour feed. Cleans up any of the debris that develops and actually ensures that you have clean cultures through those days off. And one other thing I want to note about this protocol, you may notice in the cartoon that Friday or day 5 feed is a double volume. So you want to do, if you're using, say, a 6-well plate, generally you'd feed with 2 mils. On that Friday before the 2 days off, we would suggest going up to 4 mils. And so I mentioned that Essential 8 Flex was really built upon the Essential 8 foundation. And as a result, really anything that you're doing with Essential 8 today, you should be able to pick up in Essential 8 Flex very simply. So if we're talking about matrices, we're still going to use our general suggestion for a matrix with Essential 8 Flex as well as Essential 8 is that recombinant human vitronectin. That being said, if you're interested in using something like Geltrex, you'd expect that, and actually we've generated data to show that Essential 8 Flex is perfectly compatible there as well. In terms of dissociation reagents, really the sky's the limit. Anything you're using today is compatible with Essential 8 Flex. Our suggestion, generally speaking, is that EDTA Versine clump passaging method works beautifully, but if you're using something like Accutase, Dispace, Collagenase, some of those enzymatic dissociation reagents, they're perfectly compatible with Essential 8 Flex as well. And one other dissociation reagent that I did want to point out a little bit is single-cell passaging. So this is something I've actually recently and a few other projects fallen in love with. It really does, single-cell passaging does a really nice job of maintaining consistent cultures, consistent densities over time. And what I've found is that my cultures perform best when I'm getting consistent numbers of cells plated. I know what level of confluence to expect 2, 3, 4 days later. It's nice to get in a routine like that, and single-cell passaging does a wonderful job of helping set up that routine. Now, if you're interested in exploring this with Essential 8 or Essential 8 Flex, the reagents we would recommend to go along with that are the triple select reagent. It's similar to trypsin, but a little more gentle on your cells. And you also want to make sure that, particularly when you're using something like vitronectin as a substrate, that you pair your single-cell passaging methods with the Revitacell supplement or another ROCK inhibitor. And I bring up that Revitacell supplement in particular because it's recently developed. It's actually been optimized for pluripotent stem cells. It includes a ROCK inhibitor that is significantly more specific than that Y27632 molecule that a lot of you may be using. So if you're currently doing ROCK inhibition, and you may want to look into that Revitacell supplement. It's a beautiful reagent. I've really fallen in love with it. And last but not least, we come to the cell banking. We've had great success using Essential 8 Flex for cell banking. We can freeze cells down in Essential 8 Flex. We've also used this PSC cryopreservation kit, and we've seen wonderful ability to take the cells out of cryopreservation and directly into Essential 8 Flex. And again, I bring up the Revitacell supplement because it's designed to limit the impact of stress on your PSCs. It's a great thing to add when you're taking your cells out of cryo. All right, so now that we've got, I think, a fairly decent foundation in what Essential 8 is, why it was developed, and in general, what it's going to enable you to do, I want to take a little bit of time to let you behind the curtain a little bit into our development process. So you can see the methods we use to understand how this novel media is performing, and actually you'll see some of the progress we made in developing it in real time. And then the second half of the talk, what I'm going to discuss is where we've put a lot of effort. And actually, if I'm being perfectly honest, we spent even longer than we took to develop the product itself. We have spent in validating its performance. There's been extensive real-time PSC culture testing to ensure that these cultures are high quality. And so I'm going to take some time towards the end of the talk to run through those steps. But first, I do want to get you in the right mindset for some of the media development that we went through. So as I've mentioned a few times now, we went into this program knowing that FGF2 does lose activity through exposure at 37C. And so we had a nice system set up where we would supplement various Essential 8 based formulations with FGF2. We could incubate it, say, overnight or 24 hours at 37C. And we saw a significant drop in the activity. We could measure that actually two different ways, and you're looking at an example here of how we do those measurements. In the red, you see our cell sensor assay. That's a cell-based, it's a HEC293 system, uses a FRET reporter that directly responds to the presence of active FGF2. In the blue, you're looking at an ELISA, and you can see the skew. It's actually an Invitrogen ELISA you can find on thermofisher.com. That does a really nice job of not just quantifying FGF, but actually quantifying bioactive FGF2. You can see the strong correlation between the cell sensor assay and the ELISA. But what you're looking at here as you move from left to right in this plot is actually our attempts at reformulation. So you can see our first few cracks at reformulating Essential 8. We weren't all that successful. FGF2 is still exhibiting significant loss of activity at 37C. But as we get from up to formulations 4, 5, 6, and ultimately at 7, where we landed, which eventually became Essential 8 Flex, we see nice responses in FGF2 even after prolonged exposure at 37C. So having identified what we felt was a nice formulation in order to optimize the activity of this key molecule, in addition to the other heat-sensitive potentially components that are present, what we wanted to do was get a sense for how this might be performing with regard to other commercially available PSC culture media. And so what you're looking at here are some of the results of that experiment. What we've done is taken, in this case, 3 commercially available media, supplemented them according to the manufacturer's instructions, and then incubated them at 37°C in the absence of cells. So this is just media warmed up at 37°C for periods between 1 and 72 hours. And what we see here was actually very striking to me. I knew the FGF2 sensitivity, but I had no sense for the magnitude of just how sensitive it was. You see massive losses in FGF2 activity in as short as, well, we can measure them at as short as 1 hour. But what we saw is that FGF2 activity was dropping by 50% every 4 to 8 hours, depending on the medium you were looking at. Massive drops. I mean, another way to think about this, in the period between your breakfast and your lunch, you've lost half of the FGF2. In the period between your lunch and your dinner, another half is gone. It's a fairly striking result here, particularly in light of the fact that even if we're being as diligent as we feel like we need to be, changing PSC media every 24 hours, we are experiencing FGF levels that are as low as 5 to 10% of that when we started our cultures. So it really kind of changed how I thought about our current standard PSC media and how we're interacting with our cells today. And just as a reminder from one of my first slides, culturing cells in the absence of robust FGF2 activity, it is going to lead to that spontaneous differentiation that I know we all hate to see in those cultures. Now, if I add the Essential 8 Flex formulation, our optimized formulation, to this picture, it changes dramatically. So where these competitor medias were seeing losses, significant losses, as rapidly as 2, 4, 8 hours at 37°C, in Essential 8 Flex, the FGF2 bioactivity is upwards of 90 to 95% of where it was when it started. You can look at those half-lives again, and rather than being in 4 to 8 hours, we're talking about 300 hours in order for those FGF2 activity losses to reach 50%. And just keep in mind that 300 hours is far, far beyond where we would ever leave our PSC cultures to go. And again, as a reminder, robust FGF2 expression gives you robust, happy, healthy cells. Now, having seen this result, fairly striking result, what I wanted to look at was what would happen if we pushed the system really to and beyond its limits. So in order to do some extreme stress testing, what we took was a series of those formulation attempts that I showed a few slides ago, and supplemented them with FGF2, and then incubated them at 37°C for periods of about 48 hours. Now, this pre-stressed media that we've heated for 2 days is then used to culture PSCs for up to 3 passages in that weekend-free methodology. And what we saw actually fairly quickly, I'm showing you passage 2 here in the images, but what we see fairly rapidly is significant spontaneous differentiation in in wells where FGF2 activity is not preserved. And in the case of Essential 8 Flex cultures, where we see robust maintenance of that FGF2 activity, we have very nice looking cultures, limited spontaneous differentiation. And I do want to point out that in the case of this extreme stress test, by the time we get to the end of that weekend-free feed period, or feed-free weekend period, I should say, that media has been at 37°C for upwards of 5 days. So again, far beyond where we would ever suggest or expose our routine cultures to, but the system is still holding up quite readily. And looking at this quantitatively, we looked at TRA160 expression, a nice robust pluripotency marker towards at the end of that 3rd passage. And what we see in those Essential 8 Flex cultures, again, is robust TRA160 expression in the vast majority of cells in that culture, compared to what we saw with Formulation 1, where almost all of the TRA160 expression is eliminated after that weekend-free culture in the extreme stress test. And then one last slide before I get into the long-term PSC culture work. As I was mentioning before, when I was looking at those 72-hour heat stress experiments, the results, it really did make me think about how we're interacting with our cultures today. And if you're using your standard your standard PSC medium. In the best case, when you're feeding every 24 hours, what you should think about is that those cultures are experiencing a rapid and cyclical drop in FGF2 activity followed by a spike when you feed the cells, followed by rapid drop and spike. And this goes on and on over the course of a week or obviously over the course of the lifetime of your cultures. The case is very different when we look at essential-8 flex. So what you have here is a model of what that FGF2 activity level looks like during the course of a week. With essential-8 flex, the window in which that FGF2 activity is changing is much, much smaller. We're really dropping no lower than 90 to 95%. So, another way to think about the way that these cultures are behaving at 37C, and I think really did make me think about, the way that, like I said, we've been handling our cultures historically. Okay, and so with that, I hope I've given you some semblance of, what we were thinking when we designed this product and really how we understood at first how it was performing. But what we knew was coming is long-term PSC culture work, because obviously showing that the media is working on a basic level is important, but what's really important is that your cultures are looking good and able to support the downstream work that you're so interested in doing. So in order to verify that, we use really very much the same toolkits that you all would use to validate your culture performance. I won't harp on any of these right now, but I'm going to run through some of the results that we see and just show you some side-by-side comparisons over very long-term culture of what we see. So starting with the morphology, I start here because personally I use morphology as really my first line of defense to understand how these cultures are working. Obviously the morphology is not the only indicator, but it is a pretty good one at first to understand if your cultures are behaving the way that you expect. And so what you're looking at here on the left are standard essential-8 medium cultures. These are H9 embryonic stem cells. They've been taken out 10 passages. You see the expected nice pluripotent stem cell morphology. On the right, you see parallel cultures, really equivalent cultures, maintained in essential-8 flex with weekend-free feeding. Morphology is essentially identical. Really difficult to tell one versus the other. We see the nice tightly packed colonies. We have the right high cytoplasmic to nucleus to cytoplasmic ratio, defined borders, all the things you'd expect from your PSC cultures we're seeing in both cases. And one other thing I do want to mention is throughout the rest of the talk, I'm going to be showing essential-8 versus essential-8 flex medium cultures. In every case, the essential-8 cultures are done with your standard daily feeding methodology, and then the essential-8 flex culture has been done with that optimized weekend-free protocol that I showed before. So just remember that as you're looking at these comparisons. So having seen that the cells were comfortable in their morphology, what we wanted to look at was their growth rates. So I know that in my experience in cell culture, whether it's in pluripotent stem cells or really any other cell type, one way to get my cells to grow faster, to kind of give them a kickstart, is to feed them every day. And so when your standard workflow in PSCs is to feed every day, we wanted to understand what's happening when we take away that daily feed in the presence of essential-8 flex. So what you're looking at here is a representative example of an experiment where we've taken multiple cell lines. In this case, we've got an embryonic and an induced pluripotent line, and plated those at fairly low density on a Thursday. We fed them on Friday per that optimized weekend-free protocol. And in the meantime, we're actually monitoring real-time confluence using IncuSight Zoom instrument in incubator microscopy. In the blue curve, what you see is our essential-8 cultures, where feeds were done not only Friday morning, but also Saturday morning, Sunday morning, and then additionally Monday morning. In the red, you see our essential-8 flex growth curves, and these feeds were only done in this particular instance on Thursday morning, and Friday morning and Monday morning. And what we see is very similar growth curves between these two, really limited differences. And actually, it appears that in many cases, some of those differences may be cell line specific. So I'd encourage anybody that's interested in trying this out to kind of monitor this in real time in their cells. But in the case of the H9s, where we do see a slight difference, that difference really is no more than 2, 4, maybe 6 hours. The essential-8 flex cultures are just slightly behind the essential-8. And this is a reproducible phenomenon. This happens whether you're at passage 1 or passage 10. So it's not something that we're doing to the cells themselves. And there's also a very easy fix. Say with the H9 cells, I was doing a 1:8 split routinely. Well, I might knock that down to 1:7, maybe 1:6, and I would see those curves essentially overlap entirely. So very easy fix. We really don't expect it's going to interfere with your workflows in any way. So that was encouraging to see. So once we had an understanding that these cells were looking and growing essentially the way that we expected, we thought it was important to look over long term at the pluripotency markers that they were expressing. So as a first pass, we took a molecular probes pluripotent stem cell 4 marker ICC kit, and we monitored the expression of 4 key markers over, again, prolonged culture periods. In this case, you're looking at passage 25 cultures, but the results are consistent across the board through even prolonged culture longer than this. Now, this 4-marker kit includes 2 surface markers, TRA160 in the lower left, and you can see SSEA4 in the upper right, as well as 2 nuclear stains. You got OCT4 in the upper right and SOX2 in the lower left. But across the board, what we see here is that the expression of these markers in either essentialate or essentialate flex is very robust. We see exactly the patterns we would expect. Really looking at these side by side, you'd be hard-pressed to identify which is essentialate and which is essentialate flex, which again is encouraging. One thing we also wanted to add to this panel was to look at NANOG expression. So NANOG is not included in that particular 4-marker kit. But we do know that NANOG is really sensitive to losses in pluripotency. In some cases, you may not see it in morphology. You may not see it in some of those marker expression. But if you're losing pluripotency, you're going to see differences in NANOG expression. And again, kind of the story, I'm probably sounding like a broken record at this point. The story is very similar between these 2 culture media. You're hard-pressed to tell the difference between essentialate and essentialate flex. We get robust NANOG expression throughout the life of these cultures. Now, having seen all this, the ICC is it's a nice window, a snapshot in time of a fairly small percentage of the cells that make up your culture population. So, as a supplement to this, we wanted to look at a more quantitative method. In this case, we decided to go for TRA160 expression as measured by flow cytometry. You get a very broad panel of cells, large cell numbers, and we can look at really the entire culture all at one time. What you're looking at here, honestly, is my favorite data set of everything that we've generated. I think it encompasses both the length of time that we have used to validate the performance of this, and not to mention the fact that it's really a beautiful result. In this case, we're looking at Gibco episomal iPSCs. They've been cultured on that recombinant human vitronectin I've mentioned a few times, and passaged using our standard clump methodology via Versine. And throughout the life of this culture, between passage 1 and passage 50, we've monitored TRA160 expression. And what you're seeing here, the histograms indicative of some key time points throughout these cultures. And what I found so striking, and every time I look at it, I kind of smile, is how perfectly these 2 culture sets overlay. So whether you're talking about essentialate daily feeding in those blue curves or the essentialate flex weekend free feeding in the red curves, they are almost perfectly overlaid, which is obviously a good indication that not only are the culture systems performing well, but they're performing equivalently. So the weekend free feeding in essentialate flex is not having any detrimental effects on the quality of these cultures, at least as measured by pluripotency marker expression. Now, moving forward and continuing to get a better sense of how these culture systems are performing, we wanted to look at trilineage differentiation potential. Now, we know that maintaining cells that have good pluripotency marker expression is critical to your work, but it's by no means the end. It's really just the beginning. You're growing these cells so you can use them as tools, in many cases as cells from which you can derive any cell type. And so we had to also confirm that the essentialate flex and weekend free culture was not affecting the cell's ability to go into those 3 key lineages. And so in order to validate that, again, we use multiple methods. And what you're looking at here is the synopsis of a study that actually we repeated a number of times throughout these cultures. Essentialate and essentialate flex cultures side by side were allowed to form embryoid bodies over the course of about 4 days, at which point those embryoid bodies are plated on Geltrix-coated plates and allowed to mature in the presence of a KSR supplemented medium over the course of 2 to 3 weeks. I'm not going to get into any more detail about the methodology for this particular experiment, but if anybody's interested, certainly enter that into the chat box, and I'd be happy to either reach out to you during the call, I can answer that question, or maybe what would be better is I can send you some links for information online. We have some really nice protocols available on the web. But once you have these matured cells, you fix and you stain them, and using this molecular probes 3-germ layer immunocytochemistry kit, you can look for the presence of those key lineage markers. In this case, on the left panel, you're looking at ectodermal lineage, beta-3 tubulin, so neural-like cells. For the mesodermal lineage, we're looking at smooth muscle actin. We see a really nice example of a really spread out, enormous, smooth muscle cell with the requisite fibers you'd expect. And then in terms of endodermal lineage, we're looking at alpha-fetoprotein. Actually, in some cases, these embryoid body-derived cultures, it can be tricky to get this endodermal expression, this particular marker, but you see in this case very robust expression of the endodermal lineage markers. And one question I get throughout is, okay, well, how does Essential 8 look? And I apologize for the size of these insets, but I did want you to be able to see side by side how these Essential 8 cultures look compared to Essential 8 Flex. And what, again, broken record, we're not seeing any differences. They're in both cases, all the cultures are perfectly capable of becoming those key lineages. Now, much like ICC was a nice introduction to pluripotency marker expression, we viewed that spontaneous differentiation from EBs as an introduction. It shows us that these cells are capable of forming the lineages, but doesn't necessarily tell us about how well they are able to do so. And in order to evaluate that, we actually turned to some of the newer members of the GIBCO PSC family, namely our directed differentiation kits. So moving from top to bottom, what we've done is taken cells that were at least 10 passages in Essential 8 Flex, including that weekend-free methodology, and induced them to form either definitive endoderm cells using that first kit. You can see there, I know the flow cytometry histogram is fairly small, but we see really nice, robust, consistent expression of CXCR4, greater than 95% of the cultures. So for those of you that are interested in pancreatic cells or other gut lineages, this would be the kit for you to try. It's a beautiful system. It takes 2 days. It's incredibly easy. Moving down to the middle panel is, and I know I shouldn't be playing favorites, but this is my favorite of the 3, and I think I'll show you why in the next slide. It's the GIBCO PSC Cardiomyocyte Differentiation Kit. This kit allows you to take robust pluripotent stem cell cultures, in this case from Essential 8 Flex, and induce them to form spontaneously beating cardiomyocytes. It's just fascinating stuff. I'm actually going to show you a video on the next slide. I find it mesmerizing. Whenever I get the chance to show these, I do. But you also can see in the histogram, we see consistent expression of TNNT2, which is a nice indicator of good, robust cardiomyocyte lineages. And then finally, last but not least, is the elder statesman of these 3 directed differentiation kits. That's the GIBCO PSC Neural Induction Medium. This particular kit takes your pluripotent stem cell cultures, and you're able to derive beautiful cultures of neural stem cells for your downstream continued differentiation to various neural lineages. Again, incredibly easy, takes about a week, and you end up with well over 90% of SOX1 positive beautiful neural stem cells. So for those of you doing directed differentiations, if you haven't tried these out, I strongly encourage you to do so. They were really a lifesaver in developing the data set that we have here. So as I mentioned, I can never resist showing these cardiomyocyte videos when given the opportunity. In just a minute, I'm going to fire up this video, but what you're going to be looking at in the left panel are H9, again, ESCs, taken out to passage 10 in Essential 8. In the right panel, you'll see Essential 8 Flex cultures that were done in parallel. These cells were plated on vitronectin-coated surfaces and induced to cardiomyocytes by that kit that I mentioned prior, on the slide prior. And these videos were taken 11 days after differentiation was initiated. And so with Okay, and hopefully that worked for everybody. I know we've had some technical difficulties in the past, but as I said, those videos really do mesmerize me. I could sit there and look at the microscope all day when we've got those cardiomyocytes going. But you see those nice networks of cardiomyocytes beating in unison. That really speaks for itself that the essential-8 flex medium with the feed-free, weekend-free feeding really isn't affecting the ability for these cells to form these complex differentiations. And then finally, I come to the last data set that I'll be presenting today. That's the karyotyping. So we get this question a lot. Actually, when we were first presented this idea internally, we had a lot of questions about whether we would see stable karyotypes. So this is something that we monitored very closely over time as these cultures developed, much like we did with the PSC, the pluripotency marker expression. So again, you're looking at multiple cell lines to very long passages. We have seen no indications of abnormal karyotypes. It's been actually beautiful. Across the board, we've seen really robust, normal, healthy karyotypes. Now, obviously, for those of you working in the cell therapy space and as well as regenerative medicine, can't afford to have any genomic instability or abnormality. So this was, again, a really nice result to see, and hoping to continue this work actually out past launch to continue to validate the performance of the system. So with that, I'd like to wrap up. Hopefully you've learned a little bit about our essential-8 flex medium. Hopefully you're excited about taking back control of your PSC culture schedule. Just as a reminder, you're still talking about xeno-free, feeder-free formulation here, so all the benefits of essential-8 you get, with the added benefit of preserving the bioactivity of key components. Because we're going to be eliminating those cultures, you're going to be having to spend less time in the hood, less time taking your cells out of the incubator, and actually using less media. And then, of course, the all-important no impact on the health or pluripotency of the cultures. Certainly not least, I toyed with actually starting the talk with this, but I saved it for last. An acknowledgement slide just to make sure that I gave credit to the people that really made this work possible. Two names in particular come to mind. That would be Jeff Fine and Majida Muhammad, both of which you're looking at right now. The two of these folks generated the lion's share of the data I was able to present to you today, did a spectacular job. Couldn't have been greater working with this team. And the same goes for both the extended R&D team as well as the product development team. These projects do not happen on their own. They do not happen with just R&D members. R&D loves to think that we can put on all hats and take care of everything, but we absolutely need the contributions of the greater product development team. So to them, I will be forever grateful, and I hope that for those of you out there that are interested in trying essential-8 flex, that you'll be forever grateful as well. And so with that, I will wrap up. It looks like we've gotten a good number of questions in, so I will start to address those shortly. For those of you interested in more information, as I said, we do have actually plenty of information available on the web. I'd suggest you go to thermofisher.com, search for essential-8 flex medium. You'll see our FAQs. You'll see some of this data, and hopefully you'll learn a little bit more. Appreciate the opportunity to talk today, and with that, I'd be happy to answer any questions that might have come through.
Great, Matt. Thank you so much for a great presentation. Yes, you're right. We do have a bunch of questions, which is wonderful. So why don't we start with the first one here on the list and really to find out, what kind of details can we share on how the formulation of essential-8 flex medium has changed as compared to the current catalog, essentially, the original catalog, essentially.
Right. It's a good question. It's not one I'm surprised to hear. Every time I go out and talk to customers, that one comes up. Unfortunately, we're not able to divulge too much in the way of information about that. I think that what I've said is about all that I can say. Just know that it was developed and with the essential-8 medium in mind. And so, as I said before, all the benefits that you get from essential-8, namely the xeno-free, feeder-free, serum-free, everything free, is the case with essential-8 flex as well, not to mention the cGMP quality.
Great, thank you. The next one, asking, and this is again one I know we've heard often, is we've shown the schematic with 2 passages a week. What if we're only doing 1 passage a week, or the schedule varies? How can you adjust this to the different workflows?
Right, good question. You're right, Jen. We do get that one with some regularity. I think most people the majority do tend to be using twice weekly splits, but there are plenty of people doing once weekly. And if you fall into that camp, my first suggestion would be you could maintain that schedule and simply switch out one of those split days with a feed day. So I would maintain the 3 feed-free days through the week. You can maintain that weekend-free period. But if you just swap out one of those split days for a feed day, I think you should see nice results there.
Great. Another question, in terms of the growth dynamics from clone to clone, within a line or even between different lines, how do you see differences, or how do you manage that?
Yeah, that's a good question. So as we know, every clone is going to behave a little bit differently. So when you're doing a large number of clones in parallel, you do need to do some testing to make sure that they're growing at similar rates. That really doesn't change no matter what PSC culture medium you're using. As I showed before in one of my earlier slides, there is some potential difference line to line in how they respond to Essential 8 Flex and this adopted protocol, but those differences were very small. So my hypothesis really is going to be that you'd see similar line-to-line variability using Essential 8 Flex, no different than what you see in any other PSC media. But, and I know people probably want to hear more guidance than this, but a lot of times the best way to find out is to do the testing.
All right, great. What about adaptation, whether from Essential 8 moving into Flex or from other systems?
Yep, another really good question. So what we found is that if you're moving from Essential 8 into Essential 8 Flex, the adaptation is as simple as taking your cultures at day 1 when you're doing that split, say on Monday, and plating them in Essential 8 Flex. So you give them kind of that week to adapt, and by the time the weekend rolls around, you should be good to go on skipping that weekend feed. If there are any concerns, what you could do is a slight transition period where you could use Essential 8 Flex and do a daily feed for a week or so, but we haven't really seen a need to do that in the cell lines that we have tested. Now, if you're adapting from a different culture system entirely, that can be more complicated, and we actually do have some protocols available, and I would encourage anybody to continue to reach out. If that's a particular area of interest, hit us up in the chat box again, and I'd be happy to reach out to you individually. This is an area where I mentioned that Reviticell supplement. That can be a lifesaver, particularly if you're coming from feeder conditions and you're interested in moving into feeder-free culture. That can be fairly rough on the cells, whether you're talking about weekend-free culture or not. And so I would certainly suggest to look into the Reviticell supplement, where we do have some adaptation protocols available, and I'd be happy to share those with anybody who is interested. So again, just let us know.
All right, great. Any comments in terms of spontaneous differentiation? What levels you would expect to see and how it compares to other systems?
Yeah, I mean, what we see, obviously we hate to see any spontaneous differentiation. We see very equivalent results in Essential 8 and Essential 8 Flex, and what I would extend that to say there's very little. So these culture systems are pretty stable. But obviously you do have a one-off experiment sometimes where you do get spontaneous differentiation, particularly if you've let your cells grow over confluent. You can have these culture crashes. But I don't see any additional spontaneous differentiation with this particular product in the cell lines that we have tested.
All right. Next question that came in was about reprogramming. Have we tested this in the reprogramming workflows?
Absolutely. So that was one area that was not an initial target for us, it was therefore not included in this data set. But we actually have some almost as hot off the presses data as you can get, showing really nice compatibility with Essential 8 Flex in your reprogramming. We've got essentially equivalent results between Essential 8 and Essential 8 Flex using the Cytotune workflow. So for those of you that are familiar with Cytotune, you end up culturing your cells in the media of interest, whether it's for fibroblasts or for blood cells, over the first week of the reprogramming workflow. And then at day 7, in the case of Essential 8, you just swap in your Essential 8 medium, and you'd continue to develop those PSC cultures, PSC colonies. Well, it turns out that you can really just swap in Essential 8 Flex at day 7, maintain a weekend-free weekend-free feed system, and you get really, really nice results. There's no difference in the reprogramming efficiency in Essential 8 Flex. Excellent.
All right, another question. In terms of the FGF levels, obviously with the use of Flex, you get that stable FGF. Does this cause any issues with the differentiation protocols?
No, we've seen no issues there. So I think the results that I've shown here kind of speak for themselves. Is we get really nice differentiation really down any lineage that we want to go towards. You know, we've heard concerns about this in the past, but we really have not seen the levels of FGF in these cultures inhibiting their ability to differentiate downstream.
And obviously you showed this in all the lineages, so we wouldn't expect anything different with any cardiac progenitor cells or anything along those lines?
No, I wouldn't expect any differences there at all. I mean, we have really robust differentiation down all three lineages. No red flags have shown up at this point.
All right, great. And you had a compliment on your fantastic images. So again, thank you for a great presentation.
Well, we'll have to thank Jeff and Majida for those images.
Yes, they do beautiful work.
Yeah.
I don't see any other questions rolling in right now. So we'll give it another minute and see if any other questions come in. And as Matt said, if not, he will be happy to reach out to you individually with anything else.
Absolutely. I appreciate everybody's time today. I know we're running up on the end of the hour, so thank you for sticking with us. And last suggestion, if you have any technical sales specialists visiting your lab or anybody else affiliated with Thermo Fisher, please ask them about this particular product. If you do, those questions are probably going to get back to me or Jen, so that's going to be a way for you to directly ping us if you have any follow-up. So looking forward for you testing it, and good luck to everybody.